45 million Americans. A genetic baseline of just 200. These numbers should be impossible together. One is a quarter of the country, the other is a single room of people. But in the eyes of a geneticist, the diversity of the American founding population behaves as if it came from only 200 individuals. It is a biological blueprint that has been copied, pasted, and scaled across 4 centuries. The variants present in those 200 survived. Everything else was lost. This isn't a family tree. It is a biological constraint. Most people, when they encounter the Mayflower descendant statistic, file it under genealogical trivia, the kind of fact that matters for heritage societies and ancestry websites, but has no scientific consequence. That framing is wrong. The founder effect is not a genealogy phenomenon. It is a population genetics phenomenon with specific, measurable, biologically consequential outcomes. When a new population is established by a small founding group, the genetic diversity of that population is permanently constrained by the diversity of the founders, not just statistically, physically. In the DNA, the variants that were present in the founding group persist. The variants that were absent from the founding group cannot spontaneously appear. And when the founding group is small, specific genetic characteristics, including rare variants associated with disease, can appear at elevated frequencies across the entire descended population, not because those variants are common in the general human population, but because they happen to be present in one of the 200 people who established the genetic baseline of a nation. But this is not just a story about disease genetics or rare conditions. It is a story about who those 200 people actually were, and which 5 of them are responsible for most of what remains. The Mayflower carried 102 passengers when it departed Plymouth, England in September 1620. By the end of the first winter, half of them, 50 people, were dead. Scurvy, pneumonia, and the brutal cold had reduced the founding population to just 52 survivors. Of those 52, a significant proportion did not reproduce successfully in the new colony. The effective founding population was smaller still. Of those 52 survivors, only 26 are known to have left descendants that persisted to the present day. In the eyes of genetics, the census was 102, but the genetic engine of the colony was powered by just 24 people. The Mayflower Society, which has been tracking genealogical descent from Mayflower passengers for over a century, has identified that 5 specific passenger lineages account for a disproportionate share of modern documented descent. John Alden and Priscilla Mullins, who married each other in the colony, have over 3 million documented living descendants between them. William Brewster, the colony's religious leader, Edward Winslow, Francis Cook. These 5 individuals and their spouses represent the genetic core of the Mayflower bottleneck. The estimated 35 million people worldwide with Mayflower ancestry are not descended evenly from all 102 passengers. They are descended disproportionately from a handful of super-producers who built families that exploded in size over the following decades. And before I get to what the bottleneck actually did to the genome, there is something written in the passenger demographics that deserves its own moment. Because the people on that ship were not a random sample of the English population. The Mayflower passengers were overwhelmingly drawn from 2 specific regions of England, East Anglia and the English Midlands. These were the regions with the highest concentration of separatist Puritan communities who had been subject to religious persecution under James I. They were a self-selected, geographically concentrated group. This created a pre-voyage bottleneck. They brought a genetic starter kit that was already far more homogeneous than the general English population. The founder effect didn't start in the middle of the Atlantic. It started in the local parishes of the East Midlands. But the Mayflower alone does not explain 45 million. The Mayflower was the beginning. What amplified it into a genetic phenomenon of that scale happened in the decade that followed, and it was 10 times larger. Between 1630 and 1640, approximately 20,000 Puritan settlers left England for New England in what historians call the Great Migration. Unlike the Mayflower passengers, these were not economically precarious separatists. They were intact, middle-class English families, craftsmen, merchants, minor gentry, with the economic resources to fund the crossing. And like the Mayflower passengers, they were disproportionately drawn from a narrow Puritan hearth, primarily East Anglia and the home counties surrounding London. The genetic consequence of the Great Migration is the most important amplifying event in the American founder effect story. The Mayflower established the bottleneck. The Great Migration populated it, And because the Great Migration drew from the same narrow geographic and social band as the Mayflower, the same East Anglian Puritan communities, it did not significantly broaden the genetic base. It deepened it. 20,000 people who shared not just a religion but a geographic origin, a social class, a dialect, and in many cases an overlapping network of family relationships arrived and built the genetic foundation of New England over a single decade. The 45 million Americans who carry colonial-era New England ancestry are carrying, in disproportionate measure, the genetic signature of that East Anglian Puritan founding population. But the longer I stayed with the genetics, the more I realized the Mayflower story was actually the smaller half of this. By the early 19th century, the founding families of New England had been intermarrying for 6 generations. The genetic consequence is visible in the genealogical record without any DNA analysis required. The Adams family, which produced 2 presidents, the Cabot family, the Lowell family, the Lodge family, the Winthrop family. By 1800, these were not separate lineages. They were a single extended genetic network whose members had been marrying each other's children and cousins and second cousins for generations. Oliver Wendell Holmes Sr., who coined the term Boston Brahmin in 1861, was describing a social class. He was also, without knowing it, describing a genetic bottleneck within the bottleneck, a colonial founding population that had undergone a second generation of endogamous concentration among its most powerful families. Which brings us back to the number, because an effective population of 200 is not a guess. It is a measurement. Population geneticists calculate this by looking at the reduction in genetic diversity between a source and a descendant population. When you compare the Yankee genome to its source in East Anglia, the loss of variation is staggering. The math tells us that the genetic engine of 45 million Americans behaves as if it were built from just 200 people. It is what the genome says when you ask it how many distinct genetic blueprints it was actually built from. And here's what I find most extraordinary about this specific case, because there is a community alive today that shows exactly what the Mayflower bottleneck looked like in its most extreme and most studied form. The Amish of Pennsylvania, Ohio, and Indiana are descended from approximately 200 Swiss and German Anabaptist founders who arrived in Pennsylvania between 1727 and 1750. Their community of approximately 350,000 people today traces its entire genetic ancestry back to those specific individuals. Because the Amish practice strict endogamy, marrying within the community, the founder effect has been amplified across 12 generations without any significant influx of outside genetic material. The genetic consequences in the Amish population are the most thoroughly studied example of the founder effect in any living human population. Rare genetic conditions that were present at low frequencies in the founding population, Ellis van Creveld syndrome, maple syrup urine disease, Crigler-Najjar syndrome, appear at frequencies in the Amish community that are orders of magnitude higher than in the general population, not because these conditions are caused by something specific to the Amish way of life, because the specific individuals who happened to carry these variants in the founding population of 200 had descendants who stayed in the community and married within it for 12 generations. The genetics of 1727 are still doing work in Amish bodies in 2026. The Amish example is the clearest possible demonstration of what the founder effect does at scale. But I want to stay with that word, scale, for a moment longer, because everything in this video so far has been about one founder effect, one ship, one religious community, one colonial bottleneck in one corner of one continent. And the longer I spent with the genetics of this, the more I realized I had been asking the wrong question. The founder effect did not shape the DNA of all Americans equally. It did not shape all Americans through the same bottleneck. And when you pull back far enough to see the full genetic picture of what America actually is, not the genealogical mythology, but the population genetics, what you find is not one founder effect. It is 3, running simultaneously, in the same country, none of them fully merged with the others, and each one still measurable today. The second bottleneck has nothing romantic attached to it. The transatlantic slave trade, operating from approximately 1619 to 1808, concentrated enslaved people from specific West African ethnic groups in specific American regions. The Gullah Geechee corridor of South Carolina and Georgia drawing disproportionately from Sierra Leonean Mende and Temne populations, Louisiana drawing heavily from Senegambian Wolof communities, Virginia drawing from the Bight of Biafra. The genetic consequence is a set of distinct regional founding signatures in African American DNA that are as measurable and as specific as the Mayflower bottleneck in New England colonial DNA. When researchers sequence African American mitochondrial DNA by region, the founder effect is visible in the geographic clustering. Specific haplogroups concentrate in specific counties because the slave trade, like the Mayflower, was a targeted rather than random founding event. It was not a cross-section of West Africa. It was a series of deliberate selections that concentrated specific populations in specific places and then sealed them there through the institution of slavery. The third bottleneck is 15,000 years older than either of the other two. The first Americans, the founding population whose descendants became every Native American people on the continent, crossed from Siberia into Alaska approximately 15,000 years ago. Population geneticists calculate the effective founding population of the Americas at somewhere between 100 and 1,000 individuals. Every Native American alive today, across every nation and every language and every cultural tradition on 2 continents, descends from that founding group. The founder effect in Native American DNA is the most extreme of the 3. A smaller founding population, a longer period of isolation, and a more complete genetic signature than either the Mayflower or the slave trade produced. 3 bottlenecks, 3 distinct founding events separated by thousands of years and thousands of miles. All 3 still readable in living American DNA. The founder effect is not a chapter of American history. It is the genetic architecture of the country. 3 separate compressions that built 3 distinct genetic foundations, none of which was planned, none of which was chosen by the people inside it, and all 3 of which are still doing biological work in American bodies in 2026. The bottleneck is not historical. It is biological and ongoing. The standard framing for the Mayflower founding story is nostalgic and genealogical. It is about heritage societies and Thanksgiving mythology and the romantic image of the first pilgrims building a nation from nothing. That framing treats the founder effect as a historical event, something that happened 4 centuries ago and whose consequences are now limited to the genealogical record. The genetics tells a different story. The founder effect is not a historical event. It is a biological condition that is still active in every person who carries colonial, New England founding ancestry. When a rare genetic variant associated with a specific disease appears at elevated frequency in a New England population, the explanation is not that something new happened. The explanation is that something very old happened and its consequences are still propagating through the gene pool. The bottleneck of 1620 and 1630 is not finished. It is running. In the DNA of 45 million living Americans, the effective population size of the Mayflower founding group is still constraining the range of genetic variation available to their descendants. I keep returning to what that means at the human scale. Every American who discovers through a DNA test that they carry an elevated risk for a specific condition, and who traces that condition back through their family tree to a colonial New England ancestor, is reading the biological consequence of a decision made by a specific East Anglian Puritan family in 1620 to board a ship. The genetics of that choice is still in the body, still expressing, still measurable. The 5 Mayflower passenger families who account for disproportionate modern descent, Alden, Mullins, Brewster, Winslow, Cook, were not the most prominent or powerful people on the ship. The Mayflower social hierarchy placed other families higher. What determined whose genetics dominate modern America was not status. It was survival. The families who survived the first winter found partners in the small colonial community and built large families in the subsequent decades are the families whose DNA is in 45 million living people. The most genetically consequential Americans who ever lived were not presidents or generals or founding fathers. They were people who survived one very cold Massachusetts winter in 1620 and happened to have many children. The bottleneck is not over. It is still running, in 45 million living bodies, right now. If any part of this reached you, a like costs nothing and carries more weight than most people realize for a channel doing work like this. Anyone who wants to be here the next time a story this buried gets brought into the light, subscribe and you will not miss it. Pass this to someone who has ever noticed a blank in their family's history that nobody was willing to fill in. And if a specific moment from this stayed with you, 200 survivors becoming the genetic inheritance of 45 million, 12 Amish generations still carrying the biological consequences of their founders, the bottleneck that history forgot, but the genome did not, put it in the comments. That is where the real conversation starts. Thanks for watching.